Hi, my name is Alex Rowland, also known as the Cancer Guy. I'd like to share with you an exciting new technology, the use of transcribed exosomal RNA liquid biopsies, both for detection of cancer and monitoring, and also for the potential use of designing customized treatment plans. But first off, our disclaimer. Please make sure you have read and understood the disclaimer before moving forward with this video. One of the biggest problems in cancer treatment is something called tumor heterogeneity. Heterogeneity refers to microevolution of the tumors themselves, both within, the cancer cells within a tumor and in between different tumors of the body, metastatic sites, for example. Basically, what happens is these separate tumor cells can gain or change the expression of certain genes and become resistant to treatment. So this is referred to both as spatial heterogeneity, in other words, variations within a tumor, and temporal heterogeneity over disease progression. So basically, temporal refers to the diversity over disease progression, and spatial refers to diversity between tumors within the body, which is intertumor, and diversity within a single tumor, intratumor. Liquid biopsies are the perfect solution for dealing with tumor heterogeneity. And that's because they take a snapshot of all of the tumors in the body rather than just looking at a single biopsy sample. Additionally, they can provide real-time data. In other words, they can tell you what is actually driving the cancer and metastasis today, not just from the original biopsy. So basically, we're going to look at the different types of liquid biopsies in tumors. You can have circulating tumor cells, CTCs. There's also circulating tumor DNA. And now I want to talk about circulating tumor RNA. But first, let's look at circulating tumor cells and circulating tumor DNA. So circulating tumor cells, you'll probably see these tests on the internet. There's quite a few of them. Basically, what they do is they look at something called EpCAM, which is a epithelial cell adhesion molecule that is found in early tumor cells. EpCAM is expressed in a variety of normal human epithelial tissues, carcinomas, and early stem cells. EpCAM is not found in non-epithelial skin cells or cancers of non-epithelial origin. The important thing to understand about circulating tumor cell assays is they are not specific to cancer. They can also pick up early stem cells and non-cancer cells. So early normal stem cells can also have EpCAM. Even if the test does detect actual tumor cells, only the cancer-causing stem cells can metastasize. And we know that because when they first started using animal models, they found that they had to take a tumor that had at least 6 million cells in order for it to grow and suppress the immune system. So that tells us that really only the stem cells that cause the cancer are able to metastasize and grow and spread. And the bulk of the tumor cells in a tumor will actually just die off. So cancer-causing stem cells are about 1 in 6 million cancer cells. So circulating cell-free DNA. All cells release DNA into the blood, including tumor cells. DNA in the blood is stable for quite some time. Additionally, this DNA can be isolated from the blood, and the tumor DNA can be separated from the normal DNA for quantification and qualification. However, there are various different mechanisms that cell-free DNA can be released into the blood. It can be released into the blood through apoptosis, which is programmed cell death, necrosis of a tumor cell due to treatment. It can be released through circulating tumor cells or circulating healthy cells. Also, exosomes, which are a metastatic process, release cell-free DNA and RNA and proteins and use them to spread throughout the body. We'll cover this more in detail later. So, problem with circulating tumor cells. In short, these tests identify DNA cancer-driving mutations in protein-coding genes that can be targeted with a specific drug such as BRAF V600E mutations and dabrafenib. That's the drug that targets this mutation. The hope is all of the body's tumors are driven by the same mutation, but very few tests have clinical benefits as only 2 to 3% of our genome is actually protein coding. So a small amount of tumors are driven by unique mutations such as BRAF V600E. So we'll talk a bit about the cancer genome and why this is important in detecting cancers. So the human genome contains about 2 to 3% protein coding genes. The rest of it, about 98%, is considered junk DNA, or was initially considered junk DNA. Of note, a significant part of this junk DNA is actually from retroviruses integrating into our genome over millennia. We now know that this so-called junk DNA is actually involved in regulating gene expression. Epigenetics, which is probably a term you've heard quite a bit, is actually used as a way of creating a multitude of different tissues using the same genomic template. So a quick question is, since all of our cells have the exact same DNA and genes, why do we have so many different tissues, i.e. skin, heart, muscle, etc.? And the answer is epigenetics. In short, differential gene expression results in a variety of different tissues. In other words, the genes that are turned off and on are different in every tissue. For example, cancers turn on genes that are supposed to be turned off, referred to as oncogenes, and they turn off genes that are supposed to be turned on, referred to as tumor suppressor genes. So just to summarize, we know that up to 90% of the non-coding DNA in the human genome is actually transcribed from DNA into RNA. It's not encoding proteins, so it just stays as RNA. This transcribed non-coding RNA regulates which genes are turned off and on, and for how long they are turned off and on. Importantly, one small piece of transcribed non-coding RNA can regulate thousands of genes. We also know that these transcribed non-coding RNAs get turned on years before a cancer actually occurs. Additionally, cancer cells share these transcribed non-coding RNAs with other normal cells via membrane-bound vesicles called exosomes. These exosomes are released into the blood, sweat, urine, etc., and they move around the body and get taken up by other cells. Exosomes are actually considered to be the main vehicle of both local and global metastasis. In other words, they're the way that cancer cells metastasize. So here is a bit of a background on the different mechanisms that long non-coding RNAs, which are a type of non-coding RNA, just one type, regulate different processes in cancer. So they regulate cell proliferation, cell death through apoptosis, migration, invasion, and metastasis, differentiation into normal tissues or cancer tissues, DNA damage, drug resistance, and a variety of different processes. Since these transcribed non-coding RNAs, like the previous slide of long non-coding RNAs, occupy up to 90% of our genome, why should we focus on just the 2 to 3% of protein coding DNA? Just imagine if we had a test that could capture the tumor-based exosomes from the blood and identify the various transcribed non-coding RNAs that a tumor is using to grow and spread. The predictive power of this test would allow us to determine the epigenetic drivers of metastasis, detect cancers prior to tumors being seen in imaging, reduce the amount of harmful cancer-causing radiation in scans, identify non-coding RNAs to target with drugs in real time, and also identify non-coding RNAs to target with food and supplements in real time. And they could also be used to identify non-coding RNAs for design of personalized vaccines. So, transcribed exosomal RNA liquid biopsies are actually available now. So this is a transcribed non-coding RNA liquid biopsy that we released for ASCO 2026 Gastrointestinal Symposium in San Francisco. And as you can see, the metrics are really great. Our sensitivity and specificity were really high, especially when combined with traditional imaging and blood-based biopsies. We also did one for ESMO for lung cancers, and we also had a really high sensitivity and specificity. And we're also going to be presenting at ASCO in 2026 in Chicago but I can't present that information now until it's been released publicly. So our technology, basically, what we do here is we send you 2 tubes of a specialized blood collection tube that preserves the exosomes and the RNA and DNA at room temperature. We arrange for a nurse to draw your blood. These specialized tubes preserve the exosomes at room temperature for up to a month. We then isolate the exosomes, purify the RNA, and analyze their relative expression using next generation sequencing. And we use a panel that is designed for gene expression to look at just under 21,000 transcribed RNAs and genes. We count the amount of reads per million, called RPM, and compare this to our proprietary database of normal human tissues that we have processed using the same methodology. We then look at a set of unique genes and transcribed RNAs. Currently, we're using about 405 of the 21,000 gene panel, and we know these are only overexpressed in cancers, so that's why we chose them. We can also look for additional genes or molecular features from a panel at your request. So our proprietary statistical analysis determines if these overexpressed genes or non-coding RNAs pass a predefined threshold for cancer activity based on something called the combined positive score. So the combined positive score, which uses a STAT1 and STAT2 statistics, provides us with an idea if there is cancer activity and how high that cancer activity is. This test also tells us the pathways that are involved. So we can look at the different non-coding RNAs and genes and determine which pathways are actually involved in the cancer in real time. Also, some of these exosomal transcribed RNAs can be targets of specific supplements and foods. And there's peer-reviewed data to show that certain foods can actually reduce the expression of these different genes or transcribed RNAs. Here's a classic example of different ones. Currently, we're only offering this test as a direct-to-consumer. So this means that this test is for educational purposes only and is not to be used for diagnosing, treating, or prescribing drugs for any disease whatsoever. However, this test can be used to determine and educate yourself on what molecular features are driving your cancer. And then you can use a proper diagnostic test to confirm that these genes are actually driving your cancer. So if you'd like more information on this test, you can contact us through the number here or any of our websites. Thank you.